Hello everyone, my name is Kayla, and today we will be talking about in vitro transcription of reconstituted chromatin. This video was made for MCDB 427, otherwise known as Molecular Biology at the University of Michigan. In this video, we will be discussing two experiments, but before we get into those details, I wanted to give a short review on histones and chromatin. As you may know, histones are essential eukaryotic proteins that associate with DNA in order to package it into the nucleus. When you have this DNA-histone complex, it's referred to as a nucleosome. You may have heard of this beads on a string model of chromatin, which is essentially the formation of nucleosomes that have this beaded appearance. However, many nucleosomes complex together in order to form our chromatin and our chromosomes. There are two main types of histones: core histones, such as H2A, H2B, H3, and H4, and linker histones, such as H1. The core histones are associated with the meat of our nucleosome and associate together into this large core that the DNA wraps around. H1, our linker histone, has the role to bind to linker DNA regions between our nucleosomes, which helps to stabilize our chromatin. It has actually been shown in experiments that histone H1 can be easily removed compared to the other chromosomes. Around 146 base pairs of DNA wrap around this core nucleosome, and it's common for DNA to actually have one nucleosome per 200 base pairs of DNA, which is a number that'll be important later in our experiments. Previously, evidence has been presented that histones play a role in regulating gene activity, but most molecular biologists were focusing on cracking the code of the structural importance of histones. However, one previous experiment suggested that class 3 genes in Xenopus can be selectively pressed in vitro by adding histone H1. The experiments we will be talking about today, performed by Laborn and Katanaga, wanted to show that this also held true for class 2 genes. The experimental claims that we are going to discuss today are in vitro addition of histones can lead to selective repression of specific genes, and that this repression can actually be antirepressed by activators at certain concentrations of histones. The two experiments that we are going to be discussing today, which are right here, were performed by Paul Laborn and Katanaga in 1991, and they can be described in figures 13.12 and 13.13 in Molecular Biology, 5th edition, written by Weaver. The main aspect of both of these experiments was to reconstitute or recreate chromatin in vitro. In the first experiment, Laborn and Katanaga took pure core histones and mixed it with polyglutamic acid. Now, in the actual experiment, you would have more than just 4 core histones, but for simplicity's sake, we only added 4. Polyglutamic acid is used as a good shuttle to help histones interact with DNA effectively, which is why they are mixed. We have histones in one tube and nothing yet in the other tube. Next, they added DNA, more specifically a plasmid containing the Drosophila croup gene in the presence of topoisomerase. Since we have histones and DNA, we can assume that nucleosomes are going to start forming. Of course, we're going to have many nucleosomes in our tube. Next, an in vitro transcription assay was performed with RNA polymerase and dNTPs, followed by primer extension to answer the question, do we get transcription under these conditions? Now we can take a look at the first experiment. Before we get into all of the data, we can look at all of the labels first. So at the top we can see the absence or presence of core histones, as well as polyglutamate. They also added something called sarcosyl, which just gives us one round of transcription, so you can see that right here. This is just one round. They utilized primer extension analysis, so this bracket right here with the KR is where we would expect to see our primer extension product of the croup gene that was on our plasmid. Let's take a look at the first two lanes. The first two lanes don't have any core histones added, so you can consider this naked DNA. We wouldn't have chromatin, we wouldn't have anything on the DNA, it's just our pure DNA of our plasmid. When there's no core histones and no polyglutamate, we get a decent amount of transcription right here. There's more than one band because there's more than 1+1 site. We should expect to see this because it's a eukaryotic system. Another important thing to look at is down at the bottom, there's this percent activity of transcription. Looking at our naked DNA, we can see that there's 100% activity of transcription. When polyglutamate is added, there's also 100% activity of transcription. This is just showing that polyglutamate on its own isn't affecting our transcription in any way. So looking at our lanes of naked DNA, we can conclude that high levels of transcription of our croup gene occurred. We know this because there is 100% activity, and we have some really nice bands. Now let's break up this gel and look at a certain concentration of histones. I mentioned earlier that there's often one nucleosome per 200 base pairs of DNA. In terms of our experiment, that is 0.8 ratio of histones to DNA. When we have one nucleosome per 200 base pairs, but we don't have polyglutamate, we don't get any transcription. One reason for this is if you don't have the polyglutamate to help your histones interact with the DNA, everything sort of just aggregates together and precipitates out. So you get a big mess. If you have polyglutamate and that 1 per 200 base pair, transcription goes down. We have fainter bands compared to our naked DNA lane. And an important thing to look at is our percent activity. In our naked DNA, we had 100% activity, but when we have 1 per 200 base pair of nucleosomes, we only have 24% activity, so a 75% repression, which we'll talk about later. Other than our ideal number of 1 per 200 base pair, we can also see if we increase our ratio of histones to DNA, transcription continues to decrease until we can't really detect it at all. Our overall conclusion from this figure is that adding core histones inhibits the transcription of our cripple gene as we increase the dosage of histones. One important thing we noticed in this experiment is transcription with our reconstituted chromatin when we have 1 nucleosome core per 200 base pairs of DNA caused 75% repression or only 24% activity. Why is there only that small amount of activity? Why that specific number? Let's think about what we mentioned earlier about our nucleosomes. 146 base pairs of DNA wraps around our nucleosome, and we have 1 nucleosome core per 200 base pairs. If we make it a numbers game, 146 base pairs is about 75% of our 200 base pairs of DNA per 1 nucleosome. The 50% of base pairs that's sort of out and about is 25%. There have actually been experiments performed to help model this. Let's say, for simplicity's sake, we have a piece of DNA with 1 + 1 site. If we have nucleosomes associating with this DNA, it's quite possible that one of these nucleosomes may actually block the promoter region and the +1 site of our gene. If that happens, RNA polymerase can't bind and we can't get transcription. In terms of the model, we can say this happens 75% of the time. On the other hand, if we have the same piece of DNA, we can say that the nucleosomes associate at a different region of the DNA, which can possibly leave our +1 region and our promoter region open. Therefore, RNA polymerase can bind and we can get transcription. This happens 25% of the time, and that DNA is 100% active transcriptionally. This model is saying that nucleosomes associate with the DNA randomly, and 25% of the time will leave certain promoter sequences open just due to chance. A different model states that nucleosomes associate with very specific regions of the DNA, and promoters are only 25% active overall. Based on the numbers game that we played earlier, we can sort of lean to believe this model. From our first experiment, we know that if you add core histones and reconstituted chromatin, transcription is inhibited. What about histone H1? And also, what about activators? That was the goal of the second experiment. The setup for the second experiment is very similar to our first experiment, except instead of just adding core histones, histone H1 is also added in. They wanted to see if H1 had a role in helping this repression. Again, the core histones were mixed with polyglutamic acid plus or minus H1 added in. Just like before, we'll have our histones in one reaction, but this time we're going to have H1 present. Similar to the last setup, plasmid DNA was added yet again with topoisomerase, except it's not the same plasmid as before. This time they used 2 promoter enhancer constructs, PG5E4 or PSVKR. Don't worry too much about the names right now, I'm going to describe the purpose of them in a moment. Our plasmid DNA was added to both reactions. It would either be the PSVKR, which I've shown here in blue, or the PG5E4. Nucleosomes are formed again to make our chromatin, except this time we have our linker histone. Again, you would expect to see many of these. An in vitro transcription assay was performed again, except it was in the presence or absence of certain activators, which I'll mention in the next slide. And again, primer extension was utilized to answer the question, do we get transcription under these conditions? The 2 plasmids that they utilized, PG5E4 and PSVKR, are promoter enhancer constructs. The PG5E4 construct contains 5 UAS Gal4 binding sites coupled to the adenovirus E4 promoter. You may have learned that these UAS Gal4 binding sites are binding sites for the activator Gal4. This means when PG5E4 is present in our experiment, we can use Gal4 to see if transcription is promoted in the presence of an activator. The second construct, PSVKR, is comprised of 6 GC boxes from the SV40 promoter coupled to the Drosophila croup promoter. You may have also learned that GC boxes are binding sites for the activator SP1. So yet again, when this plasmid is present, we can use SP1 to see how transcription is affected in the presence of an activator. Looking at the experiment, we can focus on the first construct first, the PG5E4 construct with those Gal4 binding sites. Looking at the labels first, there is the absence or presence of the activator Gal4VP16, there's the absence or presence of histone H1, and there's also the absence or presence of core histones. This experiment is a little different because in the first experiment we looked at different ratios of core histones to DNA, but in this experiment where there are core histones added, it's only the 0.8 histones to DNA, that 1 nucleosome per 200 base pairs of DNA. That stays the same in this experiment and the next experiment. Again, primer extension was utilized, so where this bracket is where we would expect to see our primer extension product for this plasmid. Looking at the odd lanes first, let's sort of break this gel apart into bits. Our first lane again has our naked DNA with no chromatin. And you can see that if you don't have any activator, you get a pretty basal level of transcription, just a couple bands. If we add core histones, that 1 nucleosome per 200 base pair, we can see these bands get a bit fainter. So we're having a little bit of repression. We can say the addition of core histones inhibits transcription by looking at these two lanes. On top of that, if we keep that core histone level at that 1 per 200 base pair, but we start adding concentrations of histone H1, we can see that the bands are just about gone. Transcription is decreasing. We had faint bands already with just the addition of core histones, but you can see as you add histone H1, these bands just sort of disappear. By comparing these lanes, we can see if we increase the concentration of histone H1, transcription is decreasing. Now we're going to look at the presence of activator to see what the activator can do. Again, you can see that if we add the activator, we get a lot more transcription than before. The best way to quantify this, they put at the bottom of the gel this activation. So this is the fold change in activation. Since this is naked DNA and there's nothing interfering with the activator's job, we can call this true activation by comparing these bands. Naked DNA, if you add an activator, you have an 8-fold change in activation. We can say the true activation of the Gal4 VP16 on this construct is 8-fold. Now we can look and see what our activator is doing in the presence of histones. We know that adding histones is repressing our transcription. Already looking visually, you can see that the bands are much darker when activator is present. So the activator is anti-repressing the repression that the histones caused. We can actually quantify how much anti-repression our activator is doing by comparing the total activation of that mixture at the time by the true activation of our activator on the naked DNA. For example, if we don't have any histone H1, but we have core histones, we can see that the activator is recovering this transcription by the darker bands in this lane compared to this lane. The total activation that our activator is doing in this setup is said to be 12-fold. Therefore, if we compare the total activation 12 to the true activation of 8 on the naked DNA, we can divide 12 by 8 to say that this activator in this setup antirepresses 1.5-fold. This just means that our activator is recovering 1.5 times the repression that the histones are doing. When we start adding histone H1 and core histones, we can see the activator is still recovering that transcription pretty well. In terms of this setup, it's recovering it really well with a greater than 200-fold activation. Again, we can compare the total activation, greater than 200, to the true activation 8, to say that this activator is anti-repressing 25-fold, which is pretty good. Intracellularly, you will normally have core histones as well as histone H1 in about this amount. Therefore, if you are looking at the cellular amount of anti-repression from this activator with both core and histone H1, you can expect it to be around 25-fold. Overall, we can say that if you add the Gal4 VP16 activator to this construct, the inhibition by histones is repressed. We can numerically say that the anti-repression rate is 25-fold of this activator. Now let's look at the second construct that they did, the PSV-KR construct with those GC boxes. Again, they're going to be adding or not adding the activator SP1 to see the same things that we saw in the last experiment. Let's look at our naked DNA first again, and we can see that on naked DNA, we get a decent amount of transcription. If we add core histones to this DNA, transcription is decreasing visually, we can see that. A conclusion that we can make is adding core histones inhibits transcription. We can see that by comparing these two lanes. When we start adding histone H1, we can visually see by comparing these lanes that the transcription is going down and down and down again. We can conclude from that then that as we increase the ratio of histone H1, transcription is decreasing. Now we can see what our activator is doing. Looking at the naked DNA first, we can see that if we add our activator, we're promoting a lot more transcription. It looks like a lot visually, but again, you have to look at the activation down below to get a good number from it. The true activation of this activator is only a 2.8-fold. When we start adding core histones, again, adding the activator is recovering just a little bit of our transcription. It only has a total activation of 2. Again, we can calculate this anti-repression by comparing the total activation divided by our true activation. So 2 divided by 2.8 only gives us a 0.71-fold. This number is really, really small. We can basically say that under these conditions, there isn't any anti-repression. If we start adding histone H1, however, at this 0.5 with our activator, it recovers pretty well if you just compare these two lanes visually. The number at the bottom is a 92-fold total activation. If we compare the total activation 92 to the true activation of 2.8, it's about a 33-fold anti-repression. We can sort of say that this activator is a better anti-repressor than an activator in terms of our construct. Just like in the last construct, this 0.5 is what we would expect to see cellularly with core histones as well. We can overall say that this activator has an antirepression of 33-fold. When this amount of histone H1 is starting to increase above that level, transcription is down even in the presence of our activator. We can say that the addition of the activator SP1 represses this inhibition by histone H1 and core histones. Numerically, we can say the antirepression rate is 33-fold. Overall, we can say that activators have the ability to repress the inhibitory effects that histones have on transcription. Our overall conclusions that we can see from these experiments are that class 2 genes can be repressed in vitro by adding histones. Core histones have the ability to do this on their own, but adding H1 appears to increase this effect. We can also say that activators prevent this transcription inhibition by histones by using antirepression. We can also tell that this number can vary between activators and constructs. So I hope this video was really helpful for you and you learned some really cool things about histones and activators. Happy studying and go blue!